Hello guys, welcome to my YouTube channel. So today we are going to discuss how to select and clone a full length cDNA molecule. So as you know, cDNA means complementary DNA or complete DNA from an mRNA. Okay, so the benefit of cDNA is that from a normal DNA is that the cDNA will have only the coding sequences, that is from the only exons, okay, and not the introns or the non-coding part. So whenever we are interested to look into only the coding sequences, what we do is we take the mRNA, which is obviously formed after splicing, okay, and from that mRNA we synthesize the complementary DNA. So we get only the coding sequences, and we do not get any junk or other non-coding sequences within that DNA. And we can manipulate this DNA, we can clone this DNA into a vector to synthesize our target protein. Whatever we want, we can do with that DNA. So this yellow strand that you see, remember this is the mRNA, okay, this is the mRNA. So as you know, those who have read about the structure or the function of mRNA, so you will remember that it contains a 5' guanosine cap, okay, so this is the 5' to 3' end, okay, and we give the first step is giving one primer adapter, okay. So and with this primer adapter, what else do we give, do you know? With this primer adapter, what we do is we give a restriction endonucleoside. So this is basically a primer adapter, or sometimes this is called as a hexameric sequence, or sometimes this is called as the oligo DT sequence, okay. So this is a oligo DT. Oligo DT means, oligo means some 2 to 6 bases, so these are called as oligonucleotides, and DT means these are deoxyribonucleotides, okay. So we are only giving such sequences which will have thymine, okay, as a nitrogen base, and this is a restriction site, this yellow box that you see here. So this is a restriction endonuclease site. This is a restriction endonuclease site. Got it? Then we add the reverse transcriptase enzyme. So as you know, what will a reverse transcriptase do? It will synthesize the DNA using the mRNA as a template. So as you can see here, so the DNA is synthesized from in this direction. So we know DNA is synthesized from 5' to 3' end. So this is your 5' end of the DNA, and this is your 3' end of the DNA, okay, obviously because the complementary ends of the mRNA are these and this, okay. So DNA is getting synthesized in this direction in the arrow shown here, and if you see here with this red strand which is getting synthesized, this is your DNA, okay, and the boxes that has been showed here, these are specialized nucleotides within the DNA which are called as the 5' methyl DCTP, 5' methyl DCTP, okay. Now you understand here carefully. So while you are synthesizing the DNA, you are giving all the 4 nucleotides. You are giving DCTP, DATP, DGTTP, DTP, everything you are giving, right? But after while it is getting synthesized, you have to manipulate that new strand in such a way so that all the DCTP nucleotides within the strand should get methylated, okay. Now why we are making this methylation? Because in the subsequent step you will see that we will be cutting the strand, okay, the duplex strand of the RNA DNA hybrid or the incompletely synthesized strand with RNase treatment. So because we don't want to manipulate the new strand, we don't want the new strand to get cut, okay, so that's why we are methylating one nucleotide so that the restriction enzyme, or what, not the restriction enzyme actually, the enzyme, nuclease enzyme, does not cut the DNA. It only cuts the RNA, or it separates the RNA from the DNA. Okay, so our new strand remains intact. So that's why when you are synthesizing the new strand, okay, one of the nucleotides, suppose here DCTP, should get methylated. Okay, so after this, so as you can see, in this step you will get two kinds of strand. One is, this is the first strand, is your complete strand. So complete cDNA synthesis is happening. Okay, and the next strand is your incomplete strand. So two types of strands you will be getting in this step, but we need only the complete strand. We don't need to do anything with the incomplete one. Okay, so how we are going to remove it? So that is our problem. Now what we do is, third step, we biotinylate. We biotinylate the ends of the mRNA molecule. So as you can see here, the ends of the mRNA, both the ends have been attached to biotin. Okay, so it has been attached to biotin. Now what we do is, okay, now these now we perform RNase 1 treatment. In the fourth step, what we do, we perform RNase 1 treatment. So what does RNase 1 do? RNase 1 will cleave any incomplete or single stranded portions. So as you can see here, this portion is incomplete and so it is single stranded. So this portion will be cleaved by the RNase 1. But here the complete strand does not have any incomplete or single stranded portion. So this portion will not be cleaved by RNase 1. So this portion, single stranded portion, is only prone to treatment by RNase 1. So we treat the everything with RNase 1, and only the incompletely synthesized strand will get cleaved by RNase 1, but the complete strand will remain intact. So here as you can see, this step will give you only the complete strands, and these are the incomplete strands which got cleaved. Okay, and they do not have any strand attached to them. So you only get the complete strand. So now the fifth step means you have to attach the biotinylated molecules to streptavidin coated magnetic beads. Okay, so these, as you know, biotin has a high affinity to bind to streptavidin. So here as you can see here, the biotin probes have been attached to the streptavidin molecules. Okay, so now what is the purpose of attaching it? Now you know that your sample contains two types of molecules. One is the complete strand and the other is the incomplete strand, which has been cleaved by the RNase 1. But you need to separate both the strands, right? You in your reaction media, you need to have your only full length cDNA molecules, or the full length complete strands, full length RNA DNA hybrids. You don't want any single stranded RNA DNA hybrids, right? So for that, to separate the full length molecules from the single stranded molecules, you perform biotinylation followed by attachment to streptavidin, and you figure out and you filter out only the your desired molecules. Okay, so after you filter it out, you perform RNase H treatment. So RNase H, as you know from replication also, what happens, it cleaves RNA DNA hybrids. Okay, it separates the RNA DNA hybrids. So after RNase H treatment, what you get here is, this is your cDNA molecule that you get. Okay, but this is single stranded cDNA molecule. Okay, so as we know, DNA is a double stranded. So now the problem arises, you have synthesized the cDNA from its corresponding mRNA. Now what? Now you have to synthesize the complementary strand. So that is again another problem. So what will you do? You first like attach poly DG tail to the 3' end of the DNA. Okay, so remember, this is your 3' end and this is your 5' end. Okay, and this yellow box means the restriction site. So you attach the poly DG tail to the 3' mRNA, and now you use a primer adapter. Okay, primer adapter means, so this is a very small stretch of nucleotides. Okay, so during you need the primers for PCR, and you need the primers to serve as a like initial molecules which will get extended, and it will be, the strand will get longer and it will be extended, right? So this is the primer adapter, and with it comes some stretch of cytosine molecules. Okay, so because how will you attach this primer adapter, because you have guanosine stretches in the 3' end, so the primer adapter needs to be modified in such a way so that you have some oligo DC sequence. Okay, so these are the oligo DC sequence. So you have to make your primer adapter in such a way so that oligo DC sequence comes and attaches with the oligo DC sequence of the cDNA. Otherwise, how will you attach your primer adapter? So you have to make your primer in such a way which will have this adapter. So this green box is your primer. This green box is your primer, and this stretch of the oligo DC is your adapter. Okay, so that's why these two names are called as a primer adapter. So you have to synthesize a primer adapter in such a way. Primer you have to synthesize based on your sequence, and you have to attach some bases, some oligo DC bases at the end of the primer, such that it goes and binds to the oligo DG tail of the 3' end of the newly synthesized cDNA molecule. So now what you do is you give DNA polymerase. Okay, and you give RNase H, you give DNA ligase. So what will happen? DNA polymerase will extend this strand, right, in this direction. So your second strand got synthesized in this way, in this direction. So if you remember, this was your 5' end for the second strand, this was your 3' end, and this strand that you got synthesized here, this is your 5' end, this is your 3' end. All right, and this is one restriction site that you already added. Okay, this is one restriction site that you already added during the preparation of this cDNA molecule. And those who have done primer designing, they know that we can give restriction sites within the primer also. So in this primer you will have another restriction site. Okay, right? Yes or no? Now during the synthesis of the second cDNA strand by DNA polymerase, none of the DNTPs are methylated. So as you can see here, the lower strand which you synthesize first from the mRNA has methylated DCTP, but in the upper strand there is no methylation. Okay, no methylation, right? So RNase H removes any remaining base paired RNA, and DNA ligase joins DNA segments that were synthesized internally from bits of mRNA that escaped degradation. The oligo-DC primer adapter sequence acts as a template for DNA synthesis from the 3' OH end at the end of the poly-DG tail. So your two strands of the cDNA are done. Now you have to clone it. So the basic steps of cloning comes cleaving the strands with a particular or with two restriction enzymes. If you perform generally we use two different restriction enzymes. So you have your restriction site 1, you have restriction site 2. You cut this DNA and you insert it into a cloning vector and you get it cloned, and your target protein gets expressed. So basically what we have learned here from, there are two problems. First is you have your mRNA and it is well understood that it contains only the coding regions, exons. So that is very nice. But the problem arises when you are going to synthesize the cDNA from this mRNA. Okay, so there will be an intermediate RNA DNA hybrid, and you will not get every RNA DNA hybrid which you can work with. There will be complete RNA DNA hybrids, there will be incomplete ones which will have a single stranded extensions. Okay, so now you cannot use those strands, so you have to discard the single stranded extended hybrids and you have to work with the complete strands. So okay, so what will you do? You perform RNase 1 treatment with those will clip the incomplete strands and you will have only the complete ones. Then you have to filter out the complete ones from the incomplete ones by biotin streptavidin complexes. Okay, and after that what you get is only one strand of cDNA. So second problem comes, how will you synthesize the opposite strand of the cDNA molecule? Okay, so there you have to use the primer adapter and the polydigital concept. So the polydigital will serve to hold the primer adapter molecule in its place. Understood. So you have to attach your primer to the single stranded cDNA. How will you attach? For that you have to attach polydigital first to the 3' end of the cDNA. Then you have to manipulate your primer in such a way that your primer has one adapter molecule attached to it, which will go and bind to that polydigital. And once your primer gets attached, then you give your DNA polymerase everything DNA ligase, so it will just extend that primer, so it will serve as a template. That's the purpose. So there are two basic problems, and cDNA cloning is very conceptual and it takes a lot of time. Basically what we are doing is we are doing here RT-PCR. So if you, those who know the concept of RT-PCR, so they will in lab also, whenever you are going to synthesize cDNA, you need it for cloning or maybe for expression cloning. So you have to do RT-PCR if you isolate the mRNA by trizol treatment. Generally we do trizol treatment if you are isolating the mRNA from mammalian cell cultures. Okay, so that's it. Stay tuned for more such lectures on biotechnology.